Welcome to this presentation on cell biology. So this here is a diagram showing the different structures of the cell, and it shows the inner part of the cell with the many structures, including the cytoplasm and the contents of the cytoplasm, including the mitochondria, the microtubules, the lysosomes, the peroxisomes, the Golgi complex or apparatus, the endoplasmic reticulum, the rough endoplasmic reticulum, the nucleus, the nucleolus, plus many other contents making up the cell. Now, let's now look at the nucleus. So the nucleus contains the nucleolus, woven with what? The chromatin, and the nucleus has got a nuclear envelope, which is a double layer, the nuclear envelope. And this nuclear envelope is continuous with what? The endoplasmic reticulum. Which type? The rough endoplasmic reticulum. Now, so we're saying the nucleus is made up of the nuclear envelope, and the nuclear envelope, the nuclear pores, the nucleolus, as well as the chromatin. The nuclear envelope or the nuclear membrane is made up of two layers, the inner layer and the outer layer. The inner layer contains what? The laminins, which control cell division. So the laminins control cell division. Then it is also has a phospholipid layer. It also has a phospholipid layer. Then the outer layer contains what? Ribosomes, and it is continuous or contiguous with the what? The rough endoplasmic reticulum. That is what the outer layer has. It has got ribosomes, and it is continuous with the rough endoplasmic reticulum. Then the nuclear pores, these ones act as what? Passages for passive import of small molecules, such as what? The nucleotides and other ions. It also facilitates active transport of larger molecules, such as the RNA, as they access the nuclear membrane. Then these pores are protein in nature. They are protein pores, specialized to carry out transport. So they are protein in nature. Then the other thing that we said, this nucleus contains this chromatin, which is made up of what? DNA and positively charged histone proteins. And there are 8 different types of histone proteins involved in replication, which leads to what? Mitosis. It plays a role in protein synthesis and promotes DNA condensation, leading to the formation of what? Chromosomes. That is what chromatin does. Promotes DNA condensation, leading to the formation of chromosomes. Now, histones play a role in gene expression by tightening or loosening DNA. So methylation causes the DNA to tighten, making it highly condensed, forming heterochromatin. So heterochromatin is a type of DNA that is highly condensed due to what? Methylation. And this terminates the transcription process. Hence, highly tightened DNA is not expressed. Why? Because of the methylation that is present there. So when DNA is methylated, it cannot be expressed. Then acetylation loosens DNA, forming euchromatin. So heterochromatin is because of methylation. Methylation meaning there's an addition of a methyl group to the DNA. Then acetylation forms the euchromatin. Acetylation means adding an acetyl group to the what? To the chromatin. So acetylation loosens DNA. Methylation tightens DNA. Tightened DNA is not expressed. Loosed DNA is expressed. So most of the DNA is found in this state, in the euchromatin state, loosened or loose state. Why? Because this is a form of DNA that is what gets translated now. It's one that gets translated to form actual proteins. It is the euchromatin. Then there's also the nucleolus. So nucleolus functions to synthesize RNA. That is the different types of RNA there. rRNA, which is ribosomal RNA, the messenger RNA, and the transfer RNA. Then there are also ribosomal subunits that combine with small proteins to form what? Ribosomes. So there are ribosomal subunits that combine with small proteins to form the ribosomes. That is what the nucleolus does. Then we have the endoplasmic reticulum. So the endoplasmic reticulum, like it has been mentioned, it is continuous with the outer membrane of the nuclear envelope. As you can see, that this is the nuclear envelope, double layer. Then it has got the nuclear pores that we said these functions as transport conduits and are protein in nature. So these are continuous with what? The rough endoplasmic reticulum. So rough endoplasmic reticulum, as you can see there, it has got what we call the cystine. Yeah. So what makes this rough is because so what makes this endoplasmic reticulum, this side of the endoplasmic reticulum rough, is because of the presence of the ribosomes on the surface of the cystine. So as you can see that if you look at this side, here side we have the smooth endoplasmic reticulum. Why? Because it does not have these ribosomes on the surface of the cystine there. Now what's the function of the ribosomes or the rough endoplasmic reticulum? The rough endoplasmic reticulum, it is a site for protein synthesis, which is carried out by what? The ribosomes. Then it carries out what we call N-linked glycosylation, which is a type of post-translational modification. N-linked. The proteins are organized into vesicles and are capped with proteins called the cap2 proteins that directs these vesicles to the Golgi apparatus. So remember, when the proteins are made, there has to be further modification that happens in the Golgi complex or the Golgi apparatus. Now what directs these proteins to move from the rough endoplasmic reticulum to the Golgi apparatus are the cap2 proteins. Yeah. So organs with a lot of rough endoplasmic reticulum include the pancreas, the plasma cells, the goblet cells, and the neurons. So why? Because these are involved in the production of protein containing or proteinous compounds. Yeah. Then now we have the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum, as you can see from the nucleus there, you can see that just below this side, we have the rough endoplasmic reticulum. Then we have the smooth on this other side, which does not have ribosomes on their surfaces. So it has got this lumen and the tubules. That is the smooth endoplasmic reticulum. What is the function of the smooth endoplasmic reticulum? It is for lipid synthesis. Remember that we said for the rough endoplasmic reticulum we talked about protein synthesis, but for the smooth endoplasmic reticulum we're talking about lipid synthesis, and it uses acetyl-CoA. So it has got the cytochrome P450 enzymes which metabolizes drugs, toxins, and alcohol, making them water soluble for easy excretion. So it plays a role also in degradation or metabolism of drugs, toxins, and alcohol. Then it plays a role also in glucose release in the liver by dephosphorylation of what? Glucose 6-phosphate. That is also another function of the smooth endoplasmic reticulum. Then in muscles, it stores and releases calcium for muscle contraction. Then organs with a lot of smooth endoplasmic reticulum include the testes and the ovaries, the adrenal cortex, the liver. These are some of the structures that contain a lot of smooth endoplasmic reticulum. Then now we have other organelles such as the peroxisomes. So the peroxisomes is another type of cell organelle. What does it do? So this one is involved in various metabolic processes. especially dealing with the metabolism of fat. So as you can see there, this is our peroxisome. You can see that there's a de novo lipogenesis, long chain fatty acids, very long chain fatty acids, then branched chain fatty acids. There's also a crystalline core of oxidases and bile acid synthesis intermediate. So one, it is involved in the metabolism of fatty acids. So it is a site for both alpha and beta oxidation. Yeah, so peroxisomes are hollow and spherical structures, and the main function is to do what? Carry out fatty acid oxidation, both the alpha and beta oxidation. Then also functions in metabolism of hydrogen peroxide. It also plays a role in ethanol metabolism and also in the biosynthetic functions of what? Lipids. So fatty acid oxidation, hydrogen peroxide metabolism, ethanol metabolism, biosynthetic functions of lipids. So as has been mentioned, they carry out both alpha and beta oxidation of fatty acids. So alpha oxidation is simply the breakdown of branched chain fatty acid chains into very long chains fatty acids. That is alpha oxidation. Then beta oxidation is the breakdown of very long fatty acid chains into acetyl-CoA and a fatty acyl-CoA. That is beta oxidation. Then they also contain an enzyme known as catalase, which converts hydrogen peroxide produced during what? Beta oxidation to water. That is why we're also saying they play a role in the metabolism of what? Hydrogen peroxide, because of the enzyme catalase. Yeah. Then the acetyl-CoA produces cholesterol, which can be used to synthesize what? The bile salts and the steroid hormones. So the acetyl-CoA that is used can also be used to produce cholesterol, which can furtherly be used to produce what? Bile salts as well as steroid hormones. Then again, the same acyl-CoA that is produced. So remember, these are products of what? Beta oxidation. The acetyl-CoA that is produced as a result of beta oxidation is the one that is being used to form cholesterol, which can be used to produce bile salts and steroid hormones. Then the other product of the beta oxidation that we talked about was what? Acyl-CoA. So this acyl-CoA can be converted to phospholipids by simply adding a phosphate group to it. Yeah. And the biosynthetic function of peroxisome is similar to that of the smooth endoplasmic reticulum. It simply metabolizes the ethanol similar to the smooth endoplasmic reticulum. So the catalase enzyme catalyzes the breakdown of what? Ethanol to acetaldehyde in the presence of hydrogen peroxide, releasing water and oxygen in the process. That is the function of the catalase enzyme. Then we have also ribosomes. So the ribosomes, we have these subunits. They have the small subunit and the larger subunit, and sits on the messenger RNA for what? It is in close contact with the messenger RNA as well as the tRNA, which facilitates the actual protein synthesis. So they are made up of rRNA and proteins. So they are made up of the two subunits, that is the larger and the smaller subunits. In eukaryotic cells, the larger subunit is the 60S and the smaller subunit is a 40S. There they are found on the outer surface of the rough endoplasmic reticulum in the cytoplasm. That's why you find the ribosomes. Rough endoplasmic reticulum and in the cytoplasm. That's why you find these ribosomes. So the ribosomes are the sites for the actual protein synthesis. Cytosolic ribosomes produce enzymatic proteins for the cytoplasm. So remember we said there are two sites where you can find these ribosomes. There are those that are found on the surface of the rough endoplasmic reticulum, on the surface of the cisterns of the rough endoplasmic reticulum. Then there are those that are found suspended in the cytoplasm. So those that are found suspended in the cytoplasm, they synthesize proteins that function within the cytoplasm of the cell. So that's what we are saying. That's why we're saying the cytosolic ribosomes produce enzymatic proteins for the cytoplasm. Then the rough endoplasmic reticulum ribosomes, these ones produce membrane proteins such as lysosomes, transport proteins in the cell membrane, and also export proteins such as the hormones. So these ones tend to produce proteins that are functioning as membrane proteins, that is the organelles within the cell, also as transport proteins that are found within the same cell membrane, as well as proteins that are supposed to work elsewhere, such as the hormones. All these are coming from the ribosomes making up the rough endoplasmic reticulum. Then there's also the Golgi apparatus. So the Golgi apparatus, as you can see, has got two surfaces. It has got the transphase and the cisphase. The cisphase is what receives the incoming ribosomes. Then the transphase is now the newly formed vesicles. After post-translational modification has happened, this is where they exit from. So cisphase receives the proteins that have been produced from the ribosomes. Then the vesicles containing new proteins that have been produced. Then the transphase, after these proteins have undergone modification within the lumen and the cisterns of the Golgi apparatus, they are released as newly formed vesicles on the transphase. So it has got two faces, that is the convex side known as the cis Golgi and the concave side known as the trans Golgi. The cis Golgi is the side near the rough endoplasmic reticulum and receives primary molecules that bud off the rough endoplasmic reticulum. Then the trans Golgi is the side away from the rough endoplasmic reticulum and is the side where packaged proteins exit from, like I have just explained. So the Golgi apparatus folds proteins and adds other materials, turning them into usable forms. So the Golgi apparatus folds proteins and adds other materials, turning them into usable forms. In other words, this is where post-translational modification actually occurs. Further modification happens here after protein synthesis to make them ready and turn these proteins into usable forms or active forms that can be used. The vesicles that are budding off the Golgi apparatus are going back to the rough endoplasmic reticulum. So there are some that will proceed to go and function, the others that are going to go back to the rough endoplasmic reticulum, and these will be guided by the CAP1 protein. So remember what we had the CAP2 that were guiding these vesicles containing newly formed proteins from the rough endoplasmic reticulum to the Golgi apparatus. What was guiding those proteins were the CAP2 proteins on the surface. Now, these ones that are going back to the rough endoplasmic reticulum are guided by the CAP1 proteins. The Golgi apparatus activates proteins in three ways, making them active. One, it trims the N-linked glycosylation. Remember where the N-linked glycosylation happened from? In the rough endoplasmic reticulum, right? So that N-linked glycosylation that happened is trimmed off. That's one way of activating these proteins. Then the other way is by doing what? O-linked glycosylation. O-linked glycosylation. So O-linked is only carried out by the Golgi apparatus. It is the only one that can carry out O-linked glycosylation. Then phosphorylation on mannose sugars also happens here. And now we have the lysosomes. So the lysosomes is also another membrane-bound cell organelle. It contains hydrolytic enzymes as well as transport proteins. Now, these ones are spherical vesicles these are spherical vesicles formed by phosphorylation of the mannose sugars on glycoproteins. They are spherical vesicles that are formed by phosphorylation of what? The mannose sugars on glycoproteins. And they contain hydrolytic enzymes such as the nucleases, the proteases, the lipase, glucosidases, and phosphatases. So these ones function optimally at lower than normal pH. At lower than normal pH. That's when they function best. Their membrane has a proton ATPase pump, and they break down macromolecules. They break down molecules. Yeah. Then we have the mitochondria. So the mitochondria has got this inner membrane, the outer membrane. It has also the crystal and the mitochondrial matrix there. Now, we're saying the mitochondria has got two layers. That is the outer highly permeable membrane and the inner less permeable membrane. So the outer membrane is highly permeable, the inner membrane is less permeable. So it has got an intermembrane space with a higher concentration of what? Protons. The intermembrane space has got a higher concentration of protons. So the inner membrane invaginates to form a crystal. Then the formed crystal increases the surface area for what? For metabolic reactions happening in the mitochondria. So this is our crystal. So this is our crystal here. So these foldings and invaginations form the crystal, and it is this invaginations and folding that increases the surface area. Yeah, for metabolic reactions. Then it also has a fluid field called the matrix, which contains mitochondrial DNA and ribosomes. The mitochondrial DNA is maternal in origin. So the mitochondrial DNA here is the one that is maternal in origin. What's the function? It is a site for what? The electron transport chain. It is a site for beta oxidation of fats, the Krebs cycle, gluconeogenesis, heme synthesis, ketogenesis. It is also the origin of what? The origin for apoptosis. So apoptosis is simply programmed cell death. So all these metabolic or biochemical processes happen in the mitochondria. All these are generating ATP. So that is why we simply say the mitochondria is the powerhouse, because all these are generating power for the body. That is ATP, energy for the body. Yeah, most of these processes, of course, with the exception of maybe heme synthesis, that does not directly involve ATP, but majority of these processes involve ATP, either using it up or producing it. Yeah. Then we also have the cytoskeleton. So the cytoskeleton divided into the micro, intermediate, and the macro filaments. So there are three different types. Well, the microfilaments, the intermediate, and the macro. So the microfilaments examples include the actin filaments, which are contractile in nature and play a role in cytokinesis, phagocytosis, and red cell diapedesis and muscle contraction. So the actin filaments are examples of what? Microfilaments. Then the intermediate, so these are for anchorage and anchor cells to cells, cells to extracellular matrix, and cell to organelles. Then we also have the supposed to be macro filaments. So for the macrofilament, we have the microtubules such as the alpha and beta tubulin. So these provide intracellular transport. And then they also contain the adenine and the kinesin motor proteins. There are two proteins. The two proteins require ATP to function. They are important during cell division. They are also important for cell extension such as the cilia and the flagella. That is the microfilaments. And that marks the end of this presentation. Cheers.